So now what we're going to talk about is how we think that life started on Earth, which is kind of an interesting topic because there are a lot of interesting theories for a long time, and most of those have been disproven. So we know that about 3.5 to 4 billion years ago is when Earth started to have life. It's just the process that we are not that familiar with. So there's a couple of theories that like I said, we're going to go with the one that's the most widely accepted. But there are conferences about this stuff all the time. People are always debating about it. So it's kind of interesting. So right here is an interesting one. Up until the 19th century, which really isn't that long ago, people thought that you could have spontaneous generation. And the reason they thought that is like they would leave meat out and then all of a sudden there would be maggots on it and they'd be like, well, how did those maggots get there? They just appeared, which obviously is not how that works. So spontaneous generation was believed up through the 19th century. And then Louis Pasteur came along in 1862, and he started to do pasteurization experiments. And so he would boil broth, and then he would show them that he had killed all the microbes that were living in there, and that's why there would be no mold growing or anything like that. So when you think of things that are pasteurized, that's where that comes from. So that rejected the idea of spontaneous generation. So another term that kind of came up is that really, we know that all of the living things that are on Earth right now come through a process called biogenesis. And that means that everything that's living on Earth right now came from something that was previously living, which makes sense. However, that still doesn't explain to us how we got life on Earth to start. So there's a really good hypothesis. It has 4 parts, and we're going to go through each part and then talk about how that works. We may even touch into Bio 111 terminology, which I know everybody loves. So the 4 stages are listed right here, and we're just going to go down right away and into the explanation of those. I should point out, though, steps 1 through 3 have been proven in a lab. Step 4 so far has not. They're very, very close, but they haven't gotten there yet. Okay, so the first thing was that abiotic synthesis of small organic molecules like amino acids and nucleotides. So that was the first part, followed by those turning into polymers, followed by those self-replicating, and then those turning into their own cells. So we're going to talk about those, how we got amino acids and nucleotides first. So Oparin and Haldane were going to be the guys who came up with an idea of what was in the atmosphere and what was in Earth's crust at the time when we think all of this stuff happened. Then from there were 2 scientists called Miller and Urey, and they actually set up the Miller-Urey experiment to see if they could get amino acids and nucleotides to come about. So here is a picture of the Miller-Urey experiment, and what they did is they created the ocean, which they came up with ideas from Oparin and Haldane to figure out what molecules were in there at that time. They also came up with a little primitive atmosphere. And so you can see it's got water and methane and carbon monoxide, all sorts of things like that. And what they did is they heated up their little ocean and they let that water vapor go up into these pipes. And when it started to condense and come back down, they added an electrical spark, and that was supposed to signify lightning, and in that primitive atmosphere. And then they let all of that condense, and down here they actually sampled and found organic compounds like amino acids and nucleotides. So that's pretty cool that they actually got that to happen. So the big thing that was important was that electrical spark, because if you remember, to form a chemical bond, that could require energy. And so that's where they say the energy came from, and also maybe from really strong UV light, because there wasn't any atmosphere back then. And so that's how they got that to work. Now the reason this is highly debated is because when they do it, they actually have to make sure that it has a very strong reducing environment and that it's a little concentrated. And when they actually go back to what they really think was there at that time, it doesn't work. So some people say, well, then it's crap. I think it's pretty cool that they got that far, but that's your own opinion to take. That's, Okay. So now the big idea is, how do we get these amino acids to link together and form into proteins eventually? And how do we get nucleotides to form together into nucleic acids? So this is where we're going to go back to your favorite Bio 111 topic of how we form polymers from monomers. So what they did is they took those drops of water that had the amino acids and the nucleotides in them, and they dropped that water onto hot sand or clay, because that's what they think the Earth's crust was like at that time with all the sunlight. And if you were to drop those onto hot clay, what's going to happen to the water in that droplet? Hopefully you're thinking that it's going to sizzle and evaporate off. Well, there was some process that we learned about in Bio 111, and that was a dehydration synthesis. Let's see if I can find it. I know I saw it. There it is, everybody's favorite. A dehydration synthesis. So we've got all of these amino acids and nucleotides floating around, and if we take the water out of the process, then all of a sudden we can have this polymer form. So that's how they think we got proteins and nucleic acids as a result of that part of the hypothesis. So now the next part that they had to test was how it could start self-replicating. And RNA has a tendency to self-replicate if it's given the right conditions. Now the reason they chose RNA as the genetic material instead of DNA is because you need RNA to transcribe or to translate DNA, and so they think RNA came first. So what they did in the lab is they actually put RNA and exposed it with different chemicals and different elements that they thought were in the environment at that time. And when they added zinc, it would copy itself with less than 1% error. So that's pretty cool. Kind of creepy too. Sounds like a science fiction movie or something. But anyway, that's how they proved that RNA was able to self-replicate. So we've got the building blocks of life. They're joining into polymers, and now they can actually self-replicate. The last step is to actually make it into a cell. And the reason I don't have a little section here for that is because they haven't gotten that to work in a lab without a little nudge. So they can't get it to abiotically turn into a self-contained cell that functions. So there are people working on it. It's supposedly very, very close to happening, but it hasn't completely happened yet. So that's how they think life started on Earth. Now, the last little part here that we're going to talk about is just how you organize all of these different things that are living on Earth. And so originally what they came up with was a 5-kingdom system in 1969. And so those kingdoms were Monera, Protista, Plantae, Fungi, and Animalia. So going in the other direction, you know that there's animals, fungus and molds, plants. Protists are going to be things like amoebas and kelp and a couple of diseases that cause malaria and stuff like that. And then Monera. Monera was going to just be everything that was a prokaryote. So for a long time, even when I was in college, we learned about the kingdom Monera. However, when they started to do DNA analysis of stuff, they realized that Monera was a pretty diverse group. So diverse that they thought, you know what, we have to split it out. So now, instead of 5 kingdoms, Monera is split, and so we have 6 kingdoms. We have Eukarya and bacteria, and then Archaea bacteria separate from one another. You'll still see people say Monera, but it's kind of going, it's gone in that direction where Monera no longer exists, it's obsolete, and now we have bacteria and Archaea bacteria. So in the next chapter, 26, we're actually going to talk about all the kingdoms and phylums and orders and domains and stuff. So this last little part will make a little bit more sense when we talk about that.